Good morning. So, in this session we are going to talk about cleaning and sanitation. Um, Food processing involves preserving, take the raw material, convert it into edible food through pasteurization, sterilization and many other operations. But one of the important step is the cleaning and sanitation, because you want to make sure that the environment, the plant where you are processing food is free from pathogens and spoilage organisms, other chemical hazards and things of that nature. So, when you are looking at the food processing plant, there may be a variety of things you may find. Let's suppose you are processing some flour, you may kind of have a flour spread on the floor. You may have water, because food processing, we use a lot of water for cleaning, various aspects. You may have allergens, and again, like some other things maybe doesn't bother some people, but if you are allergic to some of those things, like for example nuts and things like that, then it's a concern. The employees come from home, so they may brought some contaminants, and usually when you sanitize, you use chemicals, and if you don't properly sanitize, you may have some chemical residues. Of course, the microorganisms are there, and you probably have birds and other animals in the neighborhood, so they may try to drop things and so on and so forth, and that can be a problem. So, you have to make sure when you do the cleaning and sanitation, all these things kind of taken care of. When you do the cleaning and sanitation, you want to remove the microbiological load, allergens, and then you want to make sure it's visually and functionally clean, and there are certain similar requirement for organic labeling, kosher, halal requirement. So, you have to kind of take care of all those things. Oftentimes, when you look at the regulations, they are often talk about what they call food contact surface. So, food contact surface is where the food product is kind of directly come in contact with the equipment surfaces. So, for example, if you think about heat exchangers, the interior of the heat exchanger where the food is flowing through is an example for food contact surface. Or you are cutting some fruits and vegetables on a table, that may be another example for food contact surface. So, those food contact surfaces should free from all these residues we talked earlier. So, we have to remove them, so that we have a reduce and minimize the opportunity for cross contamination, and then they also want to avoid any potential microbiological growth. So, a lot of times, for example, you think about a table or a small equipment surface, there may be small holes in there, and you may have some food residue go and deposit there along with water. That means it's a good source for microorganisms, and if you don't check this properly, they start kind of growing there, and then that's a source of contamination. So, that's something we need to worry about. And typically, when you do the cleaning and sanitation, you have to look at this in a different zone level. The zone 1 is where the actual processing takes place. So, probably this is where your pasteurization equipment may be there, or your cutting board, or your heat exchangers, all the food processing is happening here. Then zone 2 is non-food contact surface, but it's close to that, because since it's very close, it's easy for the microorganisms to get in from the zone 2 to zone 1 quite easily. So, you have to think about that. And then zone 3 is a little bit remote from process area, but potentially you can able to have this. So, for example, you can think about processing in a room, then you probably have adjacent storage rooms where you may be keeping a lot of raw materials, and then you may have warehouse, and then you have parking lot in the building. So, it is kind of go away from the processing zone. So, each one you have to do it, but the key thing is to make sure that the food contact surface, this zone is properly sanitized. When I say cleaning, it is probably maybe simple, it is an action of making something clean, but how clean is clean? So, if I have a surface, I have some dust on the table, I can just remove the dust. Visually I look at it, it may look clean, but if I look at using a microscope or some other means, there may be still microorganisms attached to it, or maybe the other chemicals may be microscopically attached to the table. So, cleaning means you have to have certain criteria for cleaning, so that it is not like, okay, for me it looks okay clean, so I am going to done with that, whereas somebody else say, okay, this is not clean enough for me. So, there should be a consistent definition, so everyone agree upon. So, that is something needs to be discussed. The other thing when you do with the process equipment, we talked about this foulant. So, for example, when you are pasteurizing milk, you probably have some protein films can be deposited. So, this is a residue collecting on the surface as a result of contact between the surface and the product. So, that means maybe if you are processing some flour, there may be a deposit of flour may be there, and it is kind of a very thin layer, not necessarily very thick one. And then you may have protein deposits, other like starch deposits, other things may be there. And so, these are the things you have to also take care of in addition to your other residues. So, this is just to give you an example, a pipeline where you may be pasteurizing milk or you are processing juice, and if you look at it, it looks visually clean outside, but the fouling probably take place inside where the product is flowing through. So, that is where you have to take care of it, or even when you have a juice flowing through, there may be some organic matter deposit there, and then that is something needs to do it. The same way, when you have a big tank like this, how are you going to clean? What needs to be done? So, this graph just to show how the fouling can progresses and influenced by two things. One, how much time you are processing your samples. The second, at what temperature you are processing. As you can probably see here, if you are processing at 138 Celsius, this is the one, you can see that the protein deposits aggregates much more and then deposit as a thin layer much quickly in even 20 minutes. Whereas, if you are processing at 127 Celsius, you can see here it takes almost like 60 minutes to reach a similar level. So, the temperature at which you are processing and what type of proteins you have or what type of things you have kind of determine how much fouling you may have during processings. Yesterday we talked about biofilms. Biofilms can be formed on the surface. Basically, it is like a small thin layer of microorganisms, and they are much more difficult to remove compared to say even some of the foulant type thing, because microorganisms going to deposits in multiple layers, and then they kind of form a colony, and then they are much more active, and it requires much more greater effort. So, you need to have a specialized cleaning and sanitation technique needs to be there. Each product, meat, dairy, vegetables, they probably going to have different sanitizing and cleaning needs, because remember each one have different type of material. So, some of them may have more lipids in them, some of them have more proteins, some of them are carbohydrates. So, you have to kind of consider some of those things. Other thing you need to consider is cleaning and sanitation. It's the best interest of the company to do it, but it's also largely dictated by the regulations. So, for example, if you are processing products for Food and Drug Administration, government products like FDA has certain jurisdiction, which we will talk later in the next presentation. They probably have certain recommendation how you do the cleaning sanitation. USDA, they may have meat products, they may have a different recommendations. Your local authorities may have a different recommendation. So, you have to kind of think about all those things and make sure you satisfy those things. So, what do you do for cleaning? Basically, you kind of use a cleaning agent like oftentimes a detergent with say water, so that you can able to clean the surface. Second thing is you probably oftentimes you can probably clean this by pumping water at room temperature, but things will be much more effective if you do this at a relatively warmer temperature, say 60 or 70 Celsius, or depending on the operation. And then again, when you are cleaning, things may be attached to the surface. So, just rinsing water through that may not remove everything. So, some type of mechanical action. So, using a brush or other means, so that you can able to remove that better. And you also need to do this for sufficient time. Like in other words, like if you probably need make sure all the foulant and microorganisms, some other things all are removed to the extent possible, part of the cleaning process. Now, is the cleaning the same as sanitation? Sanitation refers basically getting rid of the microorganisms to certain target level, whereas cleaning primarily refers to removal of the residues, your protein films, things of that nature during the process. So, the soil is clean, visible, and then you kind of clean it. On the other hand, once you clean it to the extent possible, still there is a possibility of microorganism attached to the surface, even though surface look visibly clean. So, you have to sanitize. So, for that you have to kill the microorganism or inactivate the microorganisms, and so that you can kind of do that. You have to do it for certain level. So, for example, maybe you need about 3 log reduction, or you need about 4 log reduction, depending on the type of agencies you are working with. So, you have to kind of target your sanitation treatment based on those recommendations. So, now we are talking here something called CIP. What is CIP? CIP calls for clean in place. So, what is clean in place? And what is also, there is something called COP, this is clean out of place. So, let us think about a simple jacketed vessel. It had some impeller to mix products in there. So, after use you have to clean. So, there are 2 ways you can do. One, you can just remove this impeller, go to a sink, and you can kind of wash it, you can kind of remove it, all the things. You wash this container, and then you can put them back, assemble them, and then you kind of have it ready. So, that is the example for clean up out place. So, you have to dismantle a lot of these small parts of the equipment, and then you kind of clean outside and then assemble them, put them back. But if you ever go to a food processing plant, with so many equipment components like pasteurization or sterilization and so on. Taking equipment parts, disassemble, put them back, clean, all those things takes a lot of time. So, most of the food process operation is done what they call clean in place. So, what clean in place means that, so imagine you have a pasteurization equipment, or as an example in the class we used, you have an aseptic processing line. So, the aseptic processing line had a raw material tank, it had a pump, it had heat exchangers, it had holding tube, it has cooling heat exchanger, and then it had what I call aseptic filling, all those things. So, so it has a multi components. So, how do you disassembling all those things, put them back, probably going to take a day or two. That means your production is going to get affected. So, that means it is not going to be a very effective process. So, the idea here is you do it without disassembling by maybe passing a pre-rinse step. So, there is a sequence of steps. First, you take that pasteurization or aseptic processing equipment, then you just rinse with water. So, that any deposits of, if you are processing some soup, you may have residue soup left inside the equipment surfaces and so on. So, you kind of clean them by rinsing with water. So, it is called pre-rinse. So, this will do two things. One, if it is anything readily dissolvable, it is kind of remove them. The second thing is, in case you kind of took some time off between processing your equipment and then you start your cleaning cycle, anything dried out, it will hydrate them, so that it may kind of easy to remove in the subsequent step. The second step is your cleaning agent. You may be removing the foulant, probably adding some type of cleaning agent like detergent or something like that. So, you add some detergent to your water, and then you pass through this whole entire area again, and then you do this at a certain concentration of your detergent at certain temperature, and then you do it for a certain time. So, that you kind of want to make sure it is remove everything you can. The next step is, since you are passing through this detergent, there is going to be some detergent residue left within the equipment, and when you are going to process next time your soup again, that become a contaminant. So, you have to do is maybe do another rinse, so that you can remove your cleaning agent. Then you kind of have, maybe if you have inorganic residues, you probably do some acid cleaning. We will talk about some of the chemicals used for that later, and then you remove that acid by rinsing one more time. So, it is kind of if you look at here, food processing uses water quite a bit, because every step you start with water, and then you add some detergent, you rinse it, then you rinse it again, then you add acid, and then you rinse it again. So, this is going to be a lot of water is used. So, that is something probably later on we will talk. The technologies now being looked at is, can we do, how do I minimize the water use for the cleaning and sanitation. So, there are some efforts coming right now, but we will talk in the later slides. So, there are 4 factors affect the cleaning performance and efficiency. So, we said mechanical force. So, we talked about if you are using brushes, mechanically scrape them. So, what type of mechanical force you use, at what temperature you are sending your liquid cleaning solutions, and what is the cleaning concentration of the compounds. Are you using very dilute, or are you going to have very concentrated? So, what the appropriate concentration, and then how much contact time I have, and so on. We talked about clean out of place, and then we talked about clean in place. So, this should be CIP. So, CIP is probably what practiced in the more industrial environment. COP may be done maybe in a smaller place, where it may be much easier to manage such systems. So, manual cleaning, obviously you kind of scrub and remove the heavy soil. The second is COP, you transport the parts to operation area to a cleaning and then do this. CIP is basically you do it in the same location. So, that is kind of the 3 different places. So, again when we talk about some of the brushes, these are not very sophisticated, these are simple, but very effective in terms of doing your cleaning. So, you can kind of see ranges of brushes and other things that is commonly used. Again you can probably see here different scrapers, and you also have a drain sanitation tool, and then you can also use when you are doing cleaning, sometime what they can also do is they can apply vacuum simultaneously. So, it kind of pick up the residue much more effectively, because otherwise the material may be still remain there. So, when you select a chemical for your cleaning purpose or detergent for cleaning purpose, how do you going to select, what type of criteria do you use? I think one of the first thing you need to use is this cleaning detergent should be effective against a wide range of soil. When I say soil, these are the what I call the dirt, the foulant, the things you want to get rid of, so that you may make the surface clean. This soil does not mean that the soil you find on the earth. So, it is more a dirty kind of thing, and it should have excellent wetting and fat emulsification and sequestration properties. Remember, most of the food contain lipids, they may have carbohydrates, proteins and so on. So, that means your detergent should be able to, depending on the which detergent you use, able to take care of all those things. And you want to have this done at a low concentration. Again, especially in the current environment, the food industry is kind of trying to reduce how much chemicals you use for cleaning and sanitation. So, you do not want to use very high concentration, but you kind of try to use reasonably low concentration, so that it is still effective in cleaning, but at the same time you are not polluting the environment with lot of detergents. And water is typically used as the cleaning agent. So, the detergent should be soluble and compatible with that. And lot of the times in the plant environment, you may have hard water. So, the hard water means like how much minerals and other things you may have in your water, and is this sometime you have a luxury of getting very pure water, but in a cleaning plant environment you may not have that luxury. So, you need to be able to see whether you can able to work with wide range of water, and you should have a low foaming capacity, so that you can able to rinse without any detergent residue. So, when you have foams, the foam settle down, it may have some detergent residues. So, you want to get rid of them. Now, at the end of the day, your detergent is used in a food grade environment. So, to the extent possible, you want to have that as a food grade, so that somebody even make a mistake, at least it is reasonably forgiving than if it become a toxic or any other dyes and other things, perfumes and things of that nature. And foremost, the regulations oftentimes dictate what type of detergents you use and so on. So, here we have different type of contaminants are here, like for example, sugar, starch, protein, fat, salt and so on. And then you can probably see here how which is kind of make them solubilize. So, if you can see here, water can solubilize like sugar and salt, whereas if you look at fat, protein and starch, they are better solubilized with the alkali type of solutions. and then when you are heating these type of contaminants, what type of effect it may have, because that makes them stick together surface much more there. And then when it does, is it easy to remove or difficult to remove? So, for example, when you have protein deposits, when you heat it, they are going to denature and adhere to the surface, and it may be very difficult to remove. So, you have to use some type of alkali and hypochlorite solution, so that you can be able to remove some of these contaminants. Water is kind of used very commonly, as we said earlier, so that it is used as a pre-rinsing step, and it also transports the cleaning compound to the area to be cleaned, and then transport the suspended soil from the surface to the clean. So, water kind of functions two things, right. One, you have the cleaning agent, where you take that water and then transport to your CIP equipment. So, that is one. The second thing is, as the water is kind of water and detergent kind of wash away all the contaminants, that contaminants also going with the residue water. And you also need to rinse the cleaning compound. Finally, when you do the post rinse after your cleaning, you have to remove any residual solutions. What type of alkaline detergents are used? Typically, alkaline detergents are used to take care of organic residues, fats, oil, greases and protein. Sodium hydroxide and KOH are some of the common agents, and then hypochlorites and hydrogen peroxides, they are kind of help to remove some of the insoluble soil. Here you kind of try to highlight how appropriate levels of these detergents is needed. So, when you have detergent concentration, you can see that the axis here, time to clean, and you can see there is going to be particular concentration is going to keep the time to clean minimum. Then if you are going to increase the concentration, it probably make it more less effective. So, you have to look at what is that optimum concentration for a particular detergent. Same way, when you look at the temperature of the cleaning solution, you can probably how much time to clean. Generally, the time decreases with increase in temperature. And of course, you have to make sure that the equipment, the components of the equipment, they can tolerate the temperature, all those things. So, you have to keep that in mind. The next one is, let us say you are pumping your cleaning solution through a pipe. At what velocity you have to pump through the system. So, you can see here it is much more effective up to certain velocity, then further increase in velocity does not have much help. So, it is kind of remains about the same. So, you initially when you flow through the pipe very slow, the cleaning may not take place effectively. If you increase the speed above certain speed, then it is going to become much more effective, then suddenly it become less effective again. So, in other words, if you are trying to clean in this region is as good as doing here, but you may be spending less energy in pumping the liquid. Whereas here you are doing it at a high temperature, you are going to have much more effective cleaning. So, you rather do it here, whereas in this case somewhere in this region you may want to do it, rather than not having too little concentration or not too high concentration of the liquid. And oftentimes there are specialized devices like these type of sprinkler type mechanisms are used for cleaning difficult to clean areas. So, for example, when you have a tank, you may have just rinse the water by passing through this type of ball valve, so that it is much easier to remove. So, you have CIP solution in and CIP solution out, and you may have something like here, you can see that there is a swirling going on. So, you can able to get this done and delivered to the surface much more forcefully. So, that there is a mechanical action for cleaning. And when you do the cleaning thing, you do not want to make certain mistakes. So, for example, you may have 2 tanks. So, in this case you are pumping in the inlet CIP solution here and then get the output here and then use the same output for cleaning the second one. So, that means the second one going to have inertia to the your detergent. It also carry a lot of the soil from the first one. So, that means this may not be effective. Whereas, in this case you are going to have 2 separate lines. So, that each one going to receive its own set of detergents and it is going to be much more effective. So, how clean is clean? So, the cleaning basically you have to look at the molecular level residue occur in a certain factor. So, for example, there are some specialized things you can probably do some swab test on the surface which is being cleaned and then analyze this with certain quick methods to see how much residue may be there in ppm level or even lower. So, you can able to say okay, this is kind of acceptable level and so on. Now, when you look at sanitation, remember sanitation is a method where you typically do after you are done with the cleaning. So, you kind of first start with cleaning and after you are done with the cleaning, then you do the sanitation. And sanitation typically involve potentially inactivating any microorganisms that may be present in the surface that is being cleaned. And in the sanitation, there are 2 broad classification. One is called wet sanitation, another is we will talk later what is called dry sanitation. What is wet sanitation? As the name implies, you are using water and certain antimicrobial agents to sanitize that surface. What is dry sanitation? So, there are instances where in a food processing plant you may be processing number of dry ingredients like maybe flour, milk powder, things like that, where you do not want to use water, because in those dry processing environment if you use water can hide in the small niches and crevices, and if there is a lot of powder go and deposit there, then they serve as a source for microorganisms to grow and cause a problem. So, in those dry processing environment, they do not allow water as a processing aid or cleaning aid. So, that means you have to use a different method for sanitize them, which we will talk about that later. So, like usually the sanitizing agent, which is basically the which you use for inactivating microorganisms, they are typically chlorine based solutions, hydrogen peroxide, quaternary ammonium compounds. So, they can able to help to inactivate some of the harmful microorganisms. Now, I think from a consumer perspective, these are all considered as chemicals. So, they do have a meaningful purpose in protecting the food safety of the processing plant, but consumer may think, oh, you are using chlorine or you are using some of the ammonium compounds and so on. I do not want any chemicals in my processed food, or they may worry that the residue can get into the food. So, again that is where you have to use the appropriate concentration. So, you have to also use proper sanitation time, so that they can able to remove effectively, and then after this you have to rinse with the potable water, so that you can remove any excess chemicals. So, this is always the rinsing after your chemical use, either after cleaning or after sanitation is important. Then you also do some type of swab test, so that, or you can use some type of ATP test. So there may be microorganisms still there. So ATP test basically going to be able to say, are there any detectable microorganisms in the surface after it's being sanitized. So what are the examples of chemical sanitizers like chlorine, iodine, and I think each has certain benefits, but at the same time certain risk. So, for example, while chlorine compounds, they are very good germicides, they can able to quite effectively inactivate microorganisms. They are very corrosive to metals, because you are trying to clean your food processing plant surfaces, and if chlorine going to pit or corrode your surface, your equipment life going to decrease, and your equipment is very expensive. So, that means you have to replace. So, it becomes a concern. Other concern is there is a potential health and safety concerns, because if some of the chlorine residue get into the food, that can be a concern too. Now, iodine probably has different forms, like for example, it can effective against bacteria, viruses, and number of other organisms, but it can be a toxic in terms of when it is exposed. When you have iodine exposure, it may have some type of plastics, it can kind of provide some type of staining, and there are times it can cause irritation and so on and so forth as well. Quaternary ammonium compounds. So, these are all cationic surfactant, they can combine bactericidal and virucidal activity. They can deeply penetrate into the surface and good wearing agent, and they are typically used in floors and wall surfaces. So, hydrogen peroxide is another sanitizing agent. When you have, for example, in the aseptic processing, you have your polymeric films, you have to sanitize, you have to sterilize them, typically use hydrogen peroxide, so that you can able to get your surfaces sanitized and so on. Now, if you use above certain concentration like 5% or above, it can cause eye and skin irritant. And PAA, peroxylic acetic acid, probably has a pungent odor, and above certain concentration it is toxic. So, remember, when you use a sanitizer, when it kills the bacteria, chances are above certain concentration it can affect the human, or it can affect any other animals and so on. So, you have to use just enough concentration to kill the bacteria, and then you have to make sure you remove any residue left over, so that it does not affect when you start processing food again. And then you have bromine, which basically focus on spores, and then you can possibly combine with the chlorine to increase the effectiveness. So, we talked about dry sanitation, like where the environment you cannot use water, and so under those conditions you may be using something called superheated steam or dry ice cleaning. So, basically what you are doing is, so let us first talk about the dry sanitation environment. When you have nuts, when you have snacks, baked goods, confection, spices, all these examples for dry sanitation plants. And when you do a wet vacuum, it is okay use water, whereas in the case of dry vacuum, you are going to just remove the surface soil, but it may not remove it does not use the water under those conditions. So, what is superheated steam? So, the dry sanitation in food processing plant is still an active area of research, because there is no solution is being found. So, basically in the superheated steam you take water and then make a steam and then you heat further. So, water boils at 100 Celsius, then you heat it further to say 250 Celsius or 300 Celsius. So, the steam water turned into water vapor. What that means is, if you take a piece of paper and then you spray steam, it is probably going to get wet, because the paper is going to condense, trap the steam, and then it is going to condense on the surface and it get wet. On the other hand, when you have the superheated steam, it has a lot of latent heat. So, when you pass it through, it is going to burn. So, that means it has a lot of latent heat, that means the water vapor is not going to deposit. So, there is no deposit on water deposit on the surface. So, it behaves like a gas, but it is 100% water, you do not use chemicals, and there is no condensation after use, and you can use it to inactivate various microorganisms. So, I think the research done at Ohio State and some other Cornell and other universities suggests that this can be one potential solution for sanitizing dry sanitation plants. So, it just shows here what we already talked about. You take water and then you produce steam. This is what they call wet steam, it is visible, whereas in this case dry steam is superheated steam is going through, but it is kind of not very visible, because it is high latent energy. The next one is dry ice. So, dry ice, if you remember, it is the carbon dioxide in the solid form. So, you kind of, similar to superheated steam, you apply dry ice on the surface, so that you can able to get the sanitation done, because carbon dioxide is a good bactericidal agent. So, you can able to achieve similar thing, but again carbon dioxide, this method may be more expensive. So, recently there has been some effort where they are taking certain alcohol and then using some type of plasma technique, they vaporize the alcohol and then you apply that vaporized alcohol on the surface as a sanitation, dry sanitation method. So, that is another method being looked at. So, there is a lot of new approaches being looked at, but I think you probably on the other one I have come across the literature is they took hydrogen peroxide and then try to vaporize it and then neutralize, so that it does not have any harmful effect for the humans, but use that as a potential sanitation agent. So, there is a lot of innovations taking place in this space. So, eventually the dry sanitation can also be done much more effective. So, in summary, the cleaning food surface is basically very important, and we kind of reviewed the definition for clean in place, clean out of place, and it is important to have certain concentration, temperature and mechanical action, so that you can do your effectively clean and then sanitize the surfaces. So, these are just some suggested references, and we can have some discussions. I think So, you are talking about treating carcasses,
Yeah,
right?
treating the meat with peracetic acid, that happens in the meat industry, right? So how do they get rid of the residue that would be there on the carcass?
I think there should have been some step to remove the application at some point later in the system. So, I think I'm not that familiar with that particular operation, but there should be some step they may be using.
Okay, because I think we spoke about the residual.
Right, yeah. But like anytime you add at least from a cleaning and sanitation perspective, anytime you add a cleaning detergent or sanitizing agent, it's always important to make sure you get rid of that any potential residue.
Sure, thank you.
One of the other thing is, most of us here are food scientists and engineers. Unless you get into sanitation, you probably not going to never do sanitation in your life. And what I'm trying to say is, usually what happens is when you're processing in a plant environment, once everybody's gone, then the sanitation crew come in, probably at odd hours, and they do all the cleaning and sanitation. So, generally the rest of the people may have a less appreciation for sanitation because they never seen that's in operation. But that cleaning and sanitation is very critical for the remainder of the day when you are doing processing your food, because if they don't do the sanitation cleaning properly, you can get a lot of cross contamination that result in outbreaks and things of that nature. So, you have to make sure you can do it properly. Another example I can tell you is, like we talked about chemical, removing chemical and so on. So, there is a cantaloupe, cantaloupe fruit, they normally use water and then maybe some amount of chlorine. So, there was a time a company, they were trying to get rid of chlorine because they worried people don't want chemicals in the thing. They were just using water without any chemical intervention. Then you had certain bacteria get into that, and then it resulted in outbreak, because people just thought, okay, get rid of chemicals, but they didn't thought through, in the absence of chemical, what other thing can do its job to preserve the, take care of the microorganisms. So, it's important to pay attention to some of those details.
Anyone have any other doubts? So shall we take some break?
Okay, sure. We'll reassemble after
5 minutes.
Sure.
You will need some time, or you
No, I think we can probably start. Okay, so let's start with the next one, food process regulations. When you are processing food, a lot of things you do it in the plant environment is driven by the regulations. So, today we are going to talk about some of the regulations, probably from US perspective. So, these are all US based regulations. These probably may not be applicable if you are working in an Indian food processing plant. Nevertheless, I think many regulations originate or originally developed with a science based approach. So, that means these are all common sense, right. Like a lot of things you need to do is common sense, but somebody tried to provide them as a guidance. So, those are written eventually as a regulations. So, rather than blindly following them, trying to understand why it came through, and then what is the purpose, so that you could be able to effectively utilize them. If you look at the US, we spend about $1 billion in food safety regulations. Like if you look at a lot of the food processing plants, you may have a specialist regulatory person who may be looking after whether or not we are compliant with the various things FDA or USDA or local authorities doing, so that there. But remember, the regulations are there ultimately to protect the consumers, because I think that's the purpose of regulations. They are there to protect the consumers and public safety. And these include from farm to fork. So that means there are things you need to start doing. For example, you take milk from your farm and then you pasteurize it and then send to home through supermarket. Each step there are certain regulations you need to focus on. US regulations focus on science-based decision making, and other thing from the US side, and which is probably true in India as well, you have a federal government, you have a state government, and then you have a local government. Each may have different control over your processing plant. The regulations kind of also span from farmers, food processor, distributors, importers, and others. And I think the context by which you need to also think about this is, you may be preparing a processing ingredient, or you may be preparing some solutions which may be exported to US, and if you do that, then that company may ask, is this prepared as per the existing FDA or USDA regulations. So that means you have to make sure that it's properly prepared and so on. So having a knowledge of some of these regulations, not only in the US, Europe has its own set of regulations, and India has its own set of regulations. So you have to do it, and ultimately when you are exporting a product to a country, then you need to make sure that you follow those regulations and meet those expectations. Otherwise, the country may say, okay, I am going to reject your product because it is not in compliance with our regulations and so on. So you have to kind of pay attention to the details. So what we are trying to do here is, this is primarily focused on public safety, particularly microbiological safety. So that means you kind of want to look at pathogens in raw products, toxins, contaminants. So there's a discussion yesterday that there are certain toxins, if you are allowed to proceed, they may not be killed or eliminated by some of the processing techniques. So the best place is to avoid having toxins and similar contaminants in your food systems. You may have pesticide residues, so you kind of want to take care of them, metals, allergens, and you also have, with the recent days we are living in, there may be intentional contaminants and tampering. So like bioterrorism, things like that. So you have to kind of vigilant about all these different things. And if you look at the US, there are 12 agencies which govern food processing in food, and there are 35 regulations, and then you also have a state and federal government. What gives the FDA and USDA the authority to regulate is something called Pure Food and Drug Act, which is passed by the Congress in 1906. So it's kind of started as early as 1906, the Pure Food and Drug Act, and that basically gives the FDA the authority to regulate various food processing plants. Now let's look at some of the key regulatory agencies. So again, in the US, there are multiple agencies, and each agency has an oversight. So you may have a processing plant where you may be only processing food products which are regulated by FDA, or you may have a processing plant which regulates products USDA, or it's possible you may be doing where FDA has authority over some portion of your plant and the other portion is regulated by USDA. So, you have to kind of pay attention who is controlling what and make sure that you satisfy those needs. FDA is probably one of the major regulation, and they do most of the food except meat and poultry, and they do not do bottled water, wine and so on. So, they do 80% of the food supply, they cover seafood, dairy and produce and so on and so forth. Now, with the interesting thing is with the new developments, like for example, we have different type of meats which are not necessarily meat, but it is produced in a lab grown meat. The question kind of come in, is it governed by FDA or is it governed by USDA, because these are new technologies and the regulations not caught up with them yet. So, there are some classes of products still undecided because of the new technological advances. USDA basically cover a lot of the meat products, meat, poultry, meat containing stews, pizzas, frozen foods and so on. They get their authority, Food Safety Inspection Service get authority from the Federal Meat Safety Inspection, which is passed in 1906, and then poultry product inspection, slaughter and then egg product inspection and so on. So, the regulatory agencies FDA or USDA, they cannot just simply get into a processing plant and demand I can regulate these products. So, for each of them they have to be authorized by act of Congress, so that they have the authority to do some of those oversight. EPA basically the Environmental Protection Agency, they kind of oversee the pesticide, drinking water, prevention of toxic substances waste into the processed food. The next one is the Center for Disease Control. They kind of look at the how foodborne diseases outbreaks spread at different locations and so on, and they kind of also do surveillance and so on. With the internet available, what we see is that they have a better opportunity to get all the outbreaks reported. In the past, some of those data were not reported properly, so they were underreporting the information, whereas in the age of internet, information can be readily reported, and then with the presence of Google and many other software, when the disease is spreading, they can able to do a better job in tracking which regions have certain disease or emerging based on the keyword search, internet search, all those things. So, they kind of have a better control over some of these things now. National Oceanic Atmosphere Administration, they do seafood fisheries and they kind of collaborate with the FDA on related tasks. So, one of the things oftentimes discussed, if you look at FDA or USDA, they do a lot of things very similar or parallel, but they probably have two major agencies and they follow different procedures. And if you are a processing plant, you are doing processing of both products in the same place, you may have to do two sets of regulations and so on. So, there has been talk about merging FDA and USDA as a single food safety agency, but that requires the act of Congress, and I think that probably did not happen before. So, they still have two separate agencies, but they kind of collaborate with each other in terms of some of the related things. So, for example, when you talk about novel processing technologies, oftentimes the FDA takes the lead in terms of review the technological advances and then ask the questions, and then they issue letter of no objections. When FDA is satisfied, then USDA may follow through for its own version of the regulations for meat type of products for that type of technology. So, like as I said, like the agencies, they don't themselves regulate. Congress has to provide them authorization, and these are all an examples of authorization Congress provided. So, for example, this is some of the original one, Food and Drug Cosmetic Act in 1906, whereas if you look at some of the recent ones, Food Safety Modernization Act, I think it's about now 5-6 years, and then you have Egg Product Inspection Act and so on and so forth. So, each time a particular category of product comes in, then the Congress passes the regulations and then get into that and so on. My understanding is currently they are going through another set of regulations, so that eventually those things needs to be considered. We talked about FDA and USDA in the same processing plant, but it's also possible you may have more than FDA, USDA, but you also have a local health authority maybe in your processing plant as well. So, suppose you are doing some poultry processing, you may do some hen breeding, producing eggs, you may have USDA involved in that operation, then you are doing cleaning eggs in the processing plant. If you do it in shell, it's FDA, whereas you break open and process it, then it's USDA FSIS. Again, because the way the regulations written, if you do it in the egg, then it has to be done by FDA. You just break open and then you have the liquid egg, it may be FSIS. Then it's transported to the wholesale and then handling institutional and so on. You may have a local agriculture health departments may involve. So, you can see that the need for specialized compliance person in each processing plant overseeing different regulations. So, they may be their full time job is to look at what regulations being developed or coming up, and then try to prepare the local plant to ready for some of those things, so that the product can continue to flow without getting stuck because they are not ready for the regulations. One of the useful thing food processor find useful is called food manufacturing practices, GMP, and GMP basically define the food as the following: a food shall be deemed to be adulterated, it has been prepared, packed, held under insanitary conditions, whereby it may have become contaminated with filth, and it may be injurious to health. So, essentially what they are saying is, whenever you prepare food, you may have any bacteria or any other chemical contaminant, then they consider that as adulterated, and adulteration means you intentionally did this or you did this negligent, so that you got to allow that to happen. So, that means the processor is responsible for that, and then they can put you in contempt for the particular regulations. But the good manufacturing practices kind of provide a lot of common sense idea. So, they kind of try to say, give me minimum sanitary conditions. So, we talked about cleaning and sanitation earlier. So, they have some prescriptive cleaning and sanitation procedures part of the regulations, so that you can produce safe and wholesome food. And you have to, they don't expect you to do ultra clean. For example, if you are doing pharmaceutical type products, they expect even much more extreme sanitary requirement, whereas in the food processing sometimes those requirements are relatively modest. And a lot of times the FDA is kind of provides a oversight for GMP. So, in this picture it's probably not maybe clear, but you can probably see it may be a cockroach in your processing plant. So, if a federal inspector or FDA inspector walk in the plant and they see cockroaches or like flies and other things, that's a no, because they can probably cross contaminate a number of things and so on. So, here again what appears to be some type of corner in a processing plant where the surfaces became wet and it had some organic contacts and so on, you maybe have a mold growth or some biofilm, other things may be growing. Again, this is not acceptable. So, when you do some of this cleaning and sanitation, you have to make sure get rid of these obvious visible things from your system. And when you look at the facility, you have to store the equipment and materials properly. Again, these are all more common sense, but a lot of times people probably ignore those common sense because they don't have time or they are too busy doing other things. Then this is kind of left not done. You have to remove the waste or trash because you have to routinely do that, and then you have to properly maintain the road. You have to have a proper drainage, and you have to have a proper lighting, and you have to train the employees. So, for example, the employees going to the restroom, they have to properly wash their hands and they have proper dress code, all those things. And then you also need to have protection against pests. And in some areas processing plants may be open and birds can fly in and then they excrete things, and that can be unintentionally contaminate things. Like I think some of you may have heard about this. In California, they were processing a lot of leafy greens, and these leafy greens are coming from big multi acres of farm. Farm means you don't have any control over what's flying on the sky, and then they also have, it's like more animals may be coming through. So, there were some birds flying over, and there were some pigs running over the farm. So, they excrete things. So, that means those kind of crossed with your leaves, they come to the processing plant. In the processing plant, they did not do proper cleaning, and then they still attached to that leafy green. They were chopped. So, that means now they spread more places, and then it resulted in major outbreaks. And then they were spending a lot of time and effort to identify what caused the problem, how do you fix it, and so on and so forth. So, the point is, you cannot avoid animals going to the farm, or you cannot have plants fly into your field and so on. But if you do a proper cleaning and sanitation, you could have avoided some of those contaminants spreading further and then have all those outbreaks and so on and so forth. And I think for a while, I think consumers kind of worried because they were not sure consuming the produce is safe or not safe. So, the sale of the produce kind of declined for a particular time, then they kind of take a time to restore. So, it is important you do the cleaning and sanitation and taking care of your facilities. The equipment must be designed so that the food contact surfaces are smooth and readily accessible for cleaning and sanitation. Non-contact surface must be cleaned so that extraneous matter, microbe cannot build up and contaminate there. So, we kind of talked about CIP, COP, all those things. And one other thing, probably you heard the term HACCP, hazard analysis of critical control points. So, this is something important. So, you have to kind of try to look at your hazard, and then you kind of need to find a way to prevent them, and then you need to find a way to monitor, and if you have them, you have to corrective steps to fix it, and you verify your steps are working, and then you also do record keeping and so on. So, HACCP is something important to have. So, and federal agencies have their own share of HACCP. So, for example, you have a juice HACCP, this seafood HACCP, and then you have low acid canned foods, which is by which we are processing a lot of the shelf stable products, and then the acidified foods kind of processed by itself based on some of these regulations. So, if you go to the processing plant, they may be doing certain things certain way. Part of the thing is these are all prescribed by these regulations, and then they need to make sure it's kind of satisfy those requirement. So, for example, when you have acidified product, so remember acidified product is where you take a low acid product and then you add acidified, maybe for example, you may add some lemon juice or other things, so that you reduce the pH. So, that means under the conditions, the manufacturer should employ appropriate quality control procedure, so that the finished food do not present any hazard. So, that means it is still acidified, there is no part of that is a low acid. When you have a low acid, that means it may allow the bacterial spore to survive at that cause outbreak. And they have to also make sure that the finished for pH is less than 4.6. Remember 4.6 is the classical value, where less than 4.6 Clostridium botulinum will not grow, above 4.6 it is a risk. So, you have to kind of consider that, and then you have to do frequent testing, so that you can able to maintain the pH is properly maintained. The next we are going to talk about is food additive. So, lot of times what is food additive? So, you have food and you may kind of add or directly or indirectly, and then it kind of added to your food, it becomes part of that, it is kind of called the food additive. So, you may have, for example, have certain additives or preservatives added to your food. In the same way, Congress defined radiation as an additive, because potentially when you do radiation, there may be radiolytic compounds formed. As a result, it can potentially consider as a additive. So, that kind of comes in from the food additive definition. So, where else can food additives can happen? When you have package has a lot of chemicals, and then when you have food, the food and package interact. It is possible some of the chemicals in the package can migrate into your food, and I think again that is not probably acceptable. So, you need to make sure that no migration of the material into food is taking place. When you are processing new technology, preparing new technologies. So, for example, we talked about PEF or ohmic heating, the electrodes may be pitting or it can migrate into the metal into the food, even though it may be small quantity, but above certain threshold is not acceptable. So, migration of electrode material into PEF is probably something needs to consider. Same way when you look at X-rays, UV, pulse light, these are all some of the sources of radiation sources. So, you have to pay proper attention, so that are you going to create any radiolytic compound under certain operating conditions. The next one is food labeling. So, what that means is basically Congress want the consumers to get what the food process telling they are buying. So, in other words, they do not want you to misbrand anything. So, that means the ingredient and nutrition. So, that is why there is a ingredient statement, nutrition statement. So, that means if you are going to consume that product, you are going to have certain nutritional value or you have certain ingredients. So, for example, you may be a vegetarian, but if you have animal based product, ingredient part of that, by reading the ingredient statement, you may be able to identify that. Or you may be allergic to certain allergens like milk or egg or shellfish, tree nuts, things like that. So, you probably seen a lot of the statements, it says this product is processed in a facility that also process say peanut. So, that kind of provides. So, if you are allergic to peanut, you want to stay away from that product. So, I think that is basically one of the things. The standard of identity, basically what this is, so for example, if you are going to have a particular variety of paneer, the consumers traditionally expect the paneer to be certain composition, all those things, but you do not want to create an artificial paneer or paneer made with a imitation material. So, that is where the standard of identity says, okay, if you say this is the cheese, you have these are the type of ways in which the cheese is made and so on. More recently, the question of all natural and fresh becoming more important, and with the advance of some of the minimally processed technologies, the food processors want to use the term fresh or fresh like product. So, for example, in comparison to canning, some of the newer technologies have more fresh like appearance, ingredients and so on, but FDA has not allowed use of some of those terms. The question is, what do you think is fresh? Fresh means different things to different people, right. So, what is fresh means, and what is process means. So, for example, the raisins, right. So, where is this coming from? It is a dried grapes. Is there a fresh grape, a fresh raisin there? Can you call that as a fresh raisin, or how do you define that? So, like I think this is where the FDA has not kind of made a ruling on fresh, but I think this is a debate because people trying to use specialized terms. People say pressurized. So, like for example, high pressure processing, they kind of imply this is a fresh. So, they use the term pressurized. So, again some of them are marketing terms, but I think the FDA want to kind of try to more objectively use this particular thing. And pasteurization. So, in the past the term pasteurization until probably 1998 or 2003, the term pasteurization refers to time and temperature. Whereas, I think around 2006, FDA updated the definition for pasteurization. Now, the pasteurization can be accomplished by heat, time and temperature, or it can be accomplished by pressure and certain time, or UV and certain time, and so on and so forth. So, they kind of keep updating the definition with the times. So, we kind of talk about adulteration. So, you kind of want to avoid pesticide, chemical residue which causes insafety, and then you kind of also try to avoid like any decomposed substances, filthy substances, and if you process in sanitary conditions, and if you intentionally process a product with a diseased animal, and so anytime it kind of creates not good for people, you have to make sure that FDA can come after you because you are adulterating your food. So, even though you may not know, but I think these are considered as adulteration in the food processing. So, the term fresh is in the label when it implies that food is unprocessed. That means it is a raw state and has not been frozen, subject to any form of thermal processing, any other form of preservation. But if you look at it, most of the products in the supermarket today gone through some type of processing. So, if you look at simple apple, is it apple processed? You buy in the store? Yes, because when they get the apple, they probably wash everything clean, and then they probably add a layer of wax, so that they can kind of able to extend the shelf life all those things. So, from a stricter standpoint, that is a processed apple, but even though consumer think it's a raw product. The raw apple, basically you go yourself to the tree and pick your apple and so on. And the same way with the raisin, is it you go through drying step for the grapes, then you have raisin, but you go to the supermarket, you buy a fresh packet of raisin, you think it's fresh, whereas you keep it for 5 months, you may say okay, it's old, but in reality that grape has gone through a lot of steps before it come as a raisin. So, you have to kind of understand some of those things. So, FDA probably kind of exempt some products where they can use the term fresh. So, for example, they use waxes and other coatings. So, that is probably okay. And then if you have certain pesticides used, or you can have some mild chlorine wash on produce. So, like we mentioned, when you prepare produce, you can probably sanitize with the chlorine or some type of wash step. So, they kind of still allow that type of thing happen. Now, I think if from a regulation point of view, you can still process raw foods and then the dose is less than 1 kilogram, but in practice I think nobody use irradiation because of their consumer concerns. The next one is the term GRAS. GRAS is called generally recognized as safe. So, what that means is you are using some substance historically over so many years, thousands of years, hundreds of years before the modern food processing, then FDA consider them as GRAS. But it is also interesting that what is considered GRAS in one country may not be considered as a GRAS in a different country. So, for example, in India people may be using lot of spices and other ingredients over thousands of years. So, it is kind of very standard. Whereas, if you are newly introducing them in the US, they may not recognize that as a GRAS because for them it is a new ingredient. So, they have to go through some type of test to verify whether it is safe or not, and so on and so forth. So, this is where I think you have to petition the FDA to get a GRAS status, and then for that you have to do number of tests to get them approved. So, we kind of talked standard of identity, we talked about, like for example, Swiss cheese, it has certain expectations from consumers. So, you have to deliver that, so that it kind of provides the consistent expectation from consumers. Now, when you look at the new processing methods, how do you develop a pasteurization process? So, I think first you need to identify the microorganism of concern in the food. So, that means different locations that target microorganism may be different. So, for example, you may have a one strain of Listeria in India, whereas in Europe maybe a different strain, and then US a third strain. So, when you are processing food in India, you have to focus on the strain what is commonly found in Indian side, rather than using the strain found in US or Europe. And then you have to look at how much log reduction you need, and then you look at the food matrix impact, and then evaluate the efficacy of the process. Then you have to define the critical limits, and then what type of equipment and operating parameters you want to process. And then you kind of develop good manufacturing practices, and then you do your HACCP analysis and so on and so forth. So, in other words, like for example, if you want to take and prepare a PEF as a pasteurization method for pomegranate juice as an example, you have to take that various steps, look at what is present in the pomegranate juice, and then how much reduction, log reduction with the PEF, and then you have to see whether type of pomegranate juice in different seasons have any effect, and then you have to validate whether you achieve proper reduction, and then you look at the operating parameters, all those things, and then provide the documentation to FDA for approval. So, the validation can be three different things. One is the biological, simulant, and mathematical. So, biological is probably one of the most one FDA recommends. So, what that means is, for example, if you are processing a soup product and it is a neutral pH, you may have a Clostridium sporogenes or Clostridium botulinum as your target. Clostridium botulinum is the spore forming pathogen, sporogenes is the indicator organism. So, you have to kind of use them to deliver your thing, and then you say, okay, with the process it reduced the population by certain logs. The simulant. So, simulant is maybe some type of chemical which has a similar behavior or property like your pathogen or your surrogate. So, for example, in milk there are certain indicators, chemical indicators which behave similar to the pathogens. So, if you absent those chemical indicators, then it's free from pathogens as well. So, that is something people use, but that is much more challenging because you have to first demonstrate your chemical simulant has similar characteristics as your pathogen. Mathematical modeling, we talked about, showed examples for different mathematical models, but oftentimes mathematical model has to be validated by biological means before you can use it in the process settings. So, we kind of talked about some of these things again, and we can probably skip here. Next one important thing is the records. So, simple thing, for example, when you pasteurize milk or when you do canning, we talked about time and temperature. So, what they normally do is when you do the canning, there is something called chart recorder. So, the chart recorder, if you have a old canning equipment, it's going to have a circular recorder, and then there is a pen. It's going to just move around the chart recorder, and then it's going to record at any given time what is the temperature the product is being processed and so on. So, in other words, this give you proof in that particular batch of your canned product, it received 121 Celsius and above processing temperature. So, it's kind of record that information. Then after that processing done, you are going to identify that batch, and then you are going to store that chart recorder in a safekeeping probably about 3 years. Why? Because canned products have a longer shelf life. Maybe after 2 years a consumer can come in and then they can say, I got sick because I was consuming your canned product and that made me sick, so give me $1 million. So, one of the ways the consumers can or the food processor protect themselves is that they can produce the chart recorder from that particular batch and prove to the court or the regulatory authorities that I delivered proper time and temperature. So, when the can left my processing plant, it properly processed. So, this may happen somewhere at the consumer side or somebody else. So, I am not necessarily going to be responsible for that. If you do not have that record, then the food processor may be fined, or they may be expected to pay the consumers and so on. So, that is a chart recorder. The modern process equipment have what they call electronic chart recorder. So, it is like you are kind of, instead of writing on the chart, now you are digitally storing information in the computer, but they have specialized requirements. So, that means when you are processing and storing that information, there should be a way to safeguard that data, because you do not want to delete the data when the data is not suitable for you. So, it has to be kept there and then needs to be produced to the regulatory authority when there is a need. So, you probably want to maintain for various raw materials, packaging materials, finished products and all the process certifications, so that you are kind of provide the scheduled process. So, what is scheduled process? The scheduled process is basically what I talked about in the canning. So, you are kind of delivering certain time and temperature, my pH is certain characteristics, my water activity is certain characteristics and then my ingredients are the following. So, you kind of say okay, these are the things you define to the FDA or tell the FDA I am processing my cans with. Anything you change, any ingredient. So, for example, you are processing some soup and it has a lot of fat content. Now, you get an FDA issue no objection clearance for your soup. The consumer trend changes, now you create a low fat version of that soup. So, that means you have to reduce the fat and you modify the formulation. You cannot just simply produce that in the plant, you have to go back to the FDA and then present the modified formulation, get the FDA approval, then start processing it. So, that type of thing is needed, so that it protects the consumer, but at the same time it also protects the processor, something have it happen. And you normally recommend about 3 years, because I think that is the length of time the regulation asks you to keep them there. Process authority. So, in many places they have a process authority who kind of keep track of all these things and get them processed. And if you look at what is the definition for process authority, what is the qualification of process authority at the moment? There is no legal definition for process authority. I think the regulation says you have to have proper credentials, but does not say you have to have a bachelor degree or you have to have a PhD and so on. So, what that means is, as long as I am experienced and I have been in the field for say many years and you are willing to take that risk, then you call yourself as a process authority and then do those things. So, larger corporations tend to have process authority on the staff, whereas smaller companies, medium sized companies, maybe they kind of hire a process authority as a consultant to work for them and so on. So, more recently FDA kind of started using what they call PSMA, where basically with this Food Safety and Modernization Act, because the earlier food safety regulations were done at the time when only products were made in the US or many primarily in the US. With the globalization, lot of products are imported. So, it means that you may have an ingredient coming from country 1, country 2, country 3 and then assembled in country 4 and then export to a different country. So, how do you ensure your product is in compliance? So, that is where the Food Safety Modernization Act kind of comes in. And then there is also a growing population, which probably you get more seniors. So, for example, while this may not be a concern in India probably we have more than 40% or more or younger generation or young people less than 40 or 50, whereas in the US it is more seniors. So, even people with the age of 60 considered as young, because a lot of people are living like 70, 80 and 90. So, it is kind of there. So, for that reason they wanted to have this PSMA Act, so that they can make it careful. So, they kind of implement in 2011. So, they kind of want to put more ownership of food safety regulation to the processor, and then the FDA kind of provides more guidance, and you also try to put more emphasis on prevention. So, in summary, like the US regulations are very complex, but ideally they started as a consumer protection in mind, and FESMA regulations, which is the most recent FDA regulations, suggest that everyone has a role in ensuring safe food, and the regulations are catching up. So, it is not like stale, it is once it is there, maybe with the exception of the food additive petition in the radiation, mostly the regulations are catching up, and the globalization of food industry is also kind of giving challenges to the regulations. So, they have to kind of keep on updating it, and then there is a more need for education and outreach effect. So, that people pay attention to the regulation and comply with them. So, you have some suggestion reading materials. So, obviously, some of the documents are from FDA and then some other sources. So, I know like this is a little bit dry subject, but and I know no way I want to pretend myself as an expert in regulations, but I would be happy to discuss any questions.
Anyone having any questions? तुम लोग कुछ नहीं पूछ रहे हो रे.
Or if you have observations, you are welcome to share. Like I think if you have any observations about regulations, you can share.
Hello, sir. Sir, as we know that these authorities are there for creating the responsibility or creating a check on the industrial side. So, these authorities should not be held responsible for creating the awareness around the consumer, such that we were discussing about that many technology are there on which our consumer doesn't have faith in, and they are just having the observation that it is processed, Aruda symbol, which I was or we were talking about. They see and they try to avoid those things. So these authorities are responsible or to be held responsible to which extent that they should create an awareness, because creating a check is good thing, but creating an awareness about that check is also a good thing, that if we are just bringing it into your consciousness, it is not a hidden anything behind it.
Right. So, again, the purpose of the regulation is different from education, right. So, the purpose of the regulations are consumer protection. So, there are steps the processor need to do so that they are protected. So, I think that is one side. When you are talking about the ultra processed food, it is probably lack of understanding or mistrust from consumers about processed food, and that is a dialogue between food processors and maybe the consumers or even the academic participants and so on, and there is no need for regulations, because it is just a more education. So, for example, there are people trying to include ultra processed food, but there is no clear understanding on what basis they are causing and so on. They confuse the additives with processing, and then they summarily dismissing everything as harmful, but maybe there are ways you can improve upon some of those challenges. So, there are two different issues.
Yes, it's just look like similar that in agriculture sector, when we were going through the green revolution, that they mistook the opportunity of high yielding variety seeds. That's the same.
Right. So, yeah, I think anytime you kind of brings in innovation, you are always going to have a group of people have a confusion, and then it takes time to bring them on board as well.
So if we don't have any question, we can end this session, today's session here.
Okay. Thank you.